Hello, my name is Dorothy Ellis. I am a PhD student in the Department of Biostatistics at the University of Florida, and I am presenting Unraveling City-Specific Microbial Signatures and Identifying Sample Origins for the CAMDA 2020 Metagenomic Geolocation Challenge. This research is in collaboration with Runju Zhang, Dr. Alejandro Walker, and Dr. Susmita Datta. Here's an outline of the presentation. First, I'm going to go through a data description. Then I'm going to go through the methodology for our research, including the overview, bioinformatics and data preparation, feature selection, clustering, principal coordinates analysis, and machine learning methods. Finally, I will present our results and our conclusions. So for the data description, we have 23 cities in 17 countries, and that comprises the main data set. And then the mystery data set is comprised of 10 cities from 9 different countries, and these include new cities, Bogota, Krakow, Marseille, Naples, and Vienna. The green starred data here have multiple years worth of data, and the red starred data are also included in the mystery data set. So in addition to the microbial data, we also used some of the city-specific data that were provided. These included the summer and winter temperatures, the summer and winter humidity, a coastal city indicator, and a biome data. For the definitions of summer and winter, we use the months of the meteorological seasons. We got this data from NOAA. So for the northern hemisphere, winter is December, January, and February. And for the southern hemisphere, winter is June, July, and August. Similarly, for the northern hemisphere, summer is June, July, and August. And for the southern hemisphere, summer is December, January, and February. Now here's an overview of the workflow. First, we start with bioinformatics and data preparation. From this, we generate an OTU table of all samples. So we then have samples from cities 1 through N, and we put them through quality control, where we exclude samples with counts that have less than 20% of the median of counts for samples within that city. Next, we calculated the ubiquity of each feature across samples in cities 1 through N. And then we selected features which had a ubiquity of greater than 0.6 from each of these cities. And so these are species, orders, and families. Then we normalized the data, fit an elastic net regression model, and use that elastic net regression model to select features. Finally, we used PCOA to visualize the data and performed various machine learning methods on the data and used these to identify origins and also to identify city-specific data. This arrow here represents that we selected features based on the accuracy here. So we eventually determined a number of features based on a lower number of features, but still maintaining a fairly high accuracy. So for the bioinformatics and data preparation, the original data were Illumina sequenced in the FASTQ format. We performed data processing in University of Florida's HyperGator 2 high performance cluster. QIIME was used in open reference mode, and we performed quality control and selected data for operational taxonomic units. And all further data processing and analyses were conducted in R. And then finally, we normalized the data via log2 counts per million for downstream analyses. So for the feature selection, we had two methods of feature selection. First, we had a simple selection where we filtered samples of low quality and we selected microbial features which had a ubiquity higher than 0.6. And then we used an elastic net logistic regression. So we used, in addition to using the microbial data, we also used continuous or categorical city-specific data to fit an elastic net logistic regression model, and we use that to select microbial features. So we fit 28 models here, one for each city. And for each city's model, we used an indicator response and then retained features selected by a certain number of models. And then for the multi-output model, we used categorical multi-output. So to obtain categorical weather and humidity data, we used k-means clustering on the numerical city-specific data. And so to determine an optimal number of clusters, we used the R package NB-clust. So here's an example of what one of these clustering outputs looks like. So this is for summer humidity. We determined that 3 was the optimal number of clusters. And so we can see here that Doha and Denver, which are in Santiago, which are all pretty dry, are grouped together. For the clusters, we determined 4 clusters for winter temperature, 3 clusters for summer temperature, 2 clusters for winter humidity, and 3 clusters for summer humidity. Next, we also created a biome categorical variable for the multi-output model. We used a version of this variable as well within the elastic net regression, but we maintained it as this just urbanization score. So we generated this biome score by which indicates how urbanized the area surrounding each city is based on the Anthroms project definitions. So we gave a score of 1 if the neighboring biome was forested, 2 if it was rangeland, 3 cropland, 4 village, and 5 dense settlement. And then for the multi-output model, we created a binary categorical variable, which indicated that score was either greater than 11 or less than or equal to 11. So here we kind of have an example. So you can see that Denver has a fairly low urbanization score with 9 because they're surrounded by villages, croplands, and rangelands. And then Kiev has the highest score, which is 15, and a couple of cities had this 15 urbanization score because it's completely surrounded by dense settlements. So next, for the machine learning methods, we used 3 classifiers. These were random forest, support vector machine, and multi-layer perceptron. And we predicted sample origins with 5-fold cross-validation. So we obtained predictions from all runs, and we recorded the city with the highest and second highest number of votes for each sample for all runs. And then next we used a deep learning multi-output, multi-layer perceptron to predict selected city-specific data. So those were those 6 categorical variables that we discussed earlier. And then we used models based on the main data set to predict origins and city-specific data on samples from the mystery data set. And so for our results for the machine learning, we found that MLP obtained the lowest error rate, and we used a cutoff of 12 to obtain a low error rate without including too many features. So here we can see that the lowest error rate is with a cutoff of 7, which means that these features appeared in at least 7 models. So we had 139 OTUs, and they appeared in at least 7 of the models that we fit. So this has the lowest error rate, but we can see with just 44 features that we obtain a pretty low error rate without including an exorbitant number of features. So for this cutoff of 12, we retained 44 microbial features, and these were 14 species, 16 families, and 14 orders. So the top 5 features, meaning that they came up in the most models, were Bradyrhizobiaceae, Enterobacteriaceae, Enterobacterialis, Burkholderia, Rhizobialis, all of which were family or order. So below we have the results from PCOA. So here we can see the separation of cities. Here I believe this is New York City 16, which on the next slide we'll see has a 0 error rate. And we can see in this PCOA that it clusters fairly separately from the rest of the data, but a lot of the data overlaps with each other. And about 68% of the total variability of the data is explained by these first 2 axes. Next, for the results for the machine learning, we find that the error rate based on the highest vote from the multi-layer perceptron was 32.66%. The error rate based on the highest and second highest vote was 19.6%. So we can see here that New York City, Taipei, and Tokyo 16, NYC 16, and Tokyo 16 all obtained a 0 error rate, and for just the first vote. Now, if we include the first vote and the second vote, which means that the city that got the second highest number was the correct city, we can see that Illern, Lisbon, Denver, Berlin were also included here. And we see that Kiev and Doha 17 also don't perform particularly well, and I think that's Singapore. And then for the multi-output MLP, we then predicted 6 categorical city-specific data with multiple levels. These categorical variables were summer temperature, winter temperature, summer humidity, winter humidity, an indicator of coastal city, and a categorical variable which indicated the degree of urbanization of the biome. So the error rates for these were 20.1%, 28.6%, 24.6%, 21.1%, 15.6%, and 14.1%, respectively. And so this is for the main data set. Then for the mystery samples, we refit the MLP with mystery samples as a new category. Now, among the cities in the mystery data set, 5 of these were also in the main data set. So these were Hong Kong, Kiev, Taipei, Tokyo, and Zurich. And we found that error rates for these cities using the MLP were 46.7%, 72.7%, 18.818%, 38.5%, and 50% respectively. And we can see on the previous slides that Kiev, which performed poorly in the main data set, also performs poorly in the mystery data set, and Taipei, which had an error rate of 0%, has the lowest error rate for this prediction. Mystery samples had low sequencing depth, so this low sequencing depth somewhat contributes to the higher error rate for the predictions for these cities, because higher sequencing depth leads to cities leads to better predictions for our models. And then for cities not in the main data set, we predicted 56.7% of these samples not to belong to a city in the main data set. So then for the samples that did not belong to any of the cities in the main data set, we then predicted the city-specific categorical data. For example, we found that most of the cities from Krakow, Poland were predicted to have a similar climate to Berlin, which is the capital of Germany. And Krakow is about 530 kilometers from Berlin, so it's fairly close. So it was interesting that we were able to get that sort of climate result from these data. So in conclusion, for all three machine learning methods, cities with a greater sequencing depth and more samples had lower error rates. The MLP classifier performed better than the two other classifiers. The top 5 features were orders or families, and these orders and families were contained a very diverse set of bacteria species. So some of these bacteria naturally occur in soils. Some of them live on human skin. Some of them were nitrogen fixing. I believe one of the bacteria strains is one of the bacteria species that belongs to these orders and families was the bacteria which causes cat scratch fever. And then finally, we found that predicting city-specific climate data can identify patterns in cities and climates. And then we can use these data to draw inferences both from the predictions themselves. We can also use these predictions to narrow the range of possible cities. Finally, here's our references slide. And thank you very much for listening to our presentation. Please let me know if you have any questions.